Well, thanks a lot. I'm really excited today to be here and tell you about some of the work we've been doing trying to study lots of variants in cardiac ion channels. And I want to say up front that a lot of this work is kind of in various levels of completion or in progress. So it's really exciting for me to be at a meeting like this where you can hear the real cutting edge science and new methods and tips and tricks for how to do these experiments better. So I will spare you the 30-minute cardiac electrophysiology lecture and instead give you the 1-minute version. So this is the cardiac action potential. It governs the heartbeat in your cells, and that is controlled by ion channels, which control ions flowing in and out of your heart muscle cells in a rhythmic pattern. And there's a whole bunch of these ion channels that are interesting, but there's kind of some main top tier ones that are most associated with disease. And these include the 4 genes I've shown here, which are SCN5A, which is responsible for most of the start of the action potential, as well as a trio of different potassium channel genes shown here in blue, which are responsible for determining when the action potential ends. So as you might imagine, if you have a variant in one of these genes that severely disrupts its function, you can get some pretty serious disease, namely Mendelian or near Mendelian arrhythmia disorders. There's this a lot of interest in understanding these genes because they're considered actionable and reportable back to the physician and the patient if we discover a pathogenic variant in one of these genes. And the reason for that is that because these variants can cause potentially fatal disease. However, if we do identify these patients, there's certain actionable steps we can take, and that includes altered drug therapy. In some cases, we can even implant a defibrillator to avert the arrhythmia. So what we'd really love to know as we sequence the genomes of everybody in the world is actually understanding which variants in these genes are pathogenic. And unfortunately, this is a really hard challenge because these genes are very big, there's lots of different variants, and the risk for arrhythmia is spread over many, many rare variants. So with that in mind, we started a collaboration a few years ago with the Fowler group here in order to try to do deep mutational scans of some of these genes. So for the first part of my talk, I'm going to tell you about one gene in particular, SCN5A, which encodes the main cardiac sodium channel. We're actually interested in 2 different types of mutations in this gene. Both loss and gain of function variants can give rise to 2 different arrhythmia disorders, both of them potentially fatal, both of them partially avertable if we can recognize it. And although I drew it with kind of nice clean arrows, it's actually a little more complicated. There is a real big issue of incomplete penetrance and other genetic and environmental influences that modulate an individual's risk of getting an arrhythmia if they actually do have one of these variants. That being said, we do know there are hundreds of described pathogenic variants in this gene. Some of them are shown here in this structure. What we first started out doing is trying to develop an assay that could distinguish different flavors of SCN5A variants. And SCN5A is a little tough to study in this type of assay because it really likes being in the closed state. So no sodium actually goes through the channel. So what we did is we added 2 drugs which together bind the channel and bias it towards the open state. And what this does is it promotes sodium entry into the cell. However, the cell actually has a pump which pumps the sodium right back out. And so we added a 3rd drug which inhibits that pump, and now the cells are in trouble. So they have all the sodium coming into the cell through the channel. The main way they have to get rid of it is gone. So these cells actually die from sodium overload. So what we wanted to do is wanted to optimize this assay and see if we could actually distinguish different types of mutants with this drug treatment. So the way we did that is we developed a simple fluorescence readout in overexpressing our channel and hex cells in order to see what rate cells were dying when exposed to the drugs. And so if we do this experiment with exposing the cells to the drugs with wild type SCN5A, we get about 30–35% relative cell survival. So most of the cells are actually dying in this experiment. We then studied an allelic series of various mutants, and we could show that if you put in a nonsense mutant, which very clearly had no function, we got very little cell death in this experiment. We put in a partial loss of function variant that had, it actually had some cell death, but not as much as wild type. We put in a variant that had previously been studied and shown to have functional properties pretty similar to wild type. That behaved similar to wild type in our assay as well. And then finally, we put in a gain of function variant there at the bottom. And we were excited to see that that variant actually died at a faster rate or higher rate compared to wild type, which possibly suggested that we could use a single assay to identify both the loss and gain of function flavors of this protein. So with that, we decided to deploy this assay in a deep mutational scan. And because we're kind of novices to the deep mutational scanning methods, we decided to do a pilot experiment to prove to ourselves that we could kind of do this method and that it was working. And I should say that we're using HEC293 cell lines developed by Kenny Matreyek in the Fowler lab. And that really allows us to jump this library of variants in and study them. So this is the pilot region we chose because it had a series of well-described gain and loss of function variants in this small region. So here's the data that we got. We did a little bit of math to calculate a normalized functional score. These plots are pretty, hopefully pretty familiar to you from the last talk. But what we saw is we saw a pretty nice separation between our nonsense and synonymous classes, which are kind of our control groups. And then for our missense variants, which are ultimately the variants we're interested in understanding, we saw a cloud kind of centered near the wild type level, but stretching, including some loss and some gain of function, a putative loss and gain of function variants. And based on the replicates, we were able to kind of figure out which of these variants were excluded from overlap in the wild type range. So next we wanted to see if we actually got the right answer. So we did some validation, and this was by comparing the DMS scores to patch clamp electrophysiology results, which is the gold standard for measuring ion channel function, although it's notoriously low throughput. And so basically what we did is we took 6 previously published patch clamp results, and together with a little bit of new patch clamp data that we generated. And overall, we were pretty happy to see that the scores for 8 of the 9 variants were pretty concordant with the patch clamp data. The one exception was this boggling variant R1626H, which in the patch clamp studies actually had a pretty complex mix of mild gain and loss of function features, depending on which property of the protein you measure. In our experiment, it kind of came out as having a wild type like score. So clearly we're losing something by merging the complicated behavior of this 2,000 amino acid protein down to a single number, but we think we're capturing most of the essential disease classes of variants that we're interested in. So we were actually just funded to scale this up to the whole protein, which I'm really excited about. And so next we're kind of plugging and chugging now that this experiment's working, and we want to scan, this is a 2,000 amino acid protein, so we want to focus on the 4 transmembrane domains shown here. And these are where the vast majority of the pathogenic variants lie. And so we're starting with domain 4. I was hoping to have the data ready for you, but it was about a week off in terms of the analysis, so stay tuned. So for the second part of my talk, I want to switch gears a little bit and tell you about a second approach that we're doing to study cell surface abundance or cell surface trafficking. And kind of the rationale for this is that for a lot of these transmembrane proteins at the cell surface, the major mechanism of loss of function is it just never gets to the right place. So it doesn't traffic to the cell surface. So shown here as an example for KCNH2, so there's actually been about 200 variants in this protein that have been described and manually studied as loss of function. And of those, 85% of them have trafficking deficiency as the mechanism. So we are obviously not going to capture every functional feature of the protein if we study this, but we think we can capture a large percentage of the loss of function variants. And furthermore, we think this could be a potentially portable assay that can be relatively quickly applied to different transmembrane genes. So shown here is an example of how we actually do these measurements. We have a small epitope tag in an extracellular loop of the protein, such as an HA tag. And then if we stain live cells, which have an intact cell membrane with an antibody against that tag, we actually only are measuring the protein at the cell surface. And we've done validation of these tags across a whole number of genes and showed that we can get good tags that work. So what we do is we take a library of these mutants, and they presumably have differential cell surface abundance. We stain the pool of cells, and we get a staining that looks something like this, where you have some cells that don't stain at all, some cells that stain very well, and then we can flow sort and sequence these pools. So for this gene KCNH2, it's kind of a team effort, but it's spearheaded by my colleague at Vanderbilt, Brett Cronke. And so here's, again, we're kind of doing, starting with these pilot experiments just to kind of prove to ourselves we can do the method before scaling up. So here's an example pilot scan of KCNH2. an 11 amino acid region. Again, we see a really nice separation between our nonsense and synonymous classes and our missense variants. In this case, it looks like there's some variants that have a relatively normal cell surface abundance all the way down to loss of function. And so we think, and I'm not going to show you, but we've actually gone back and validated a lot of these variants manually in single cell flow cytometry or confocal experiments and shown that these scores are accurate. So here's another example. This is another gene, one of the potassium channels, KCNE1. So it actually has it's the smallest of all the genes I've told you about. So we actually made a library of the whole thing, which we're excited about. And so this is data showing that the tag is working. We can detect good cell surface expression, cell surface abundance of the wild type, as opposed to a loss of function mutant L51H, which doesn't traffic to the cell surface. If we look at our library of all the mutants, we see a distribution that overlaps the wild type distribution pretty well. However, we have a kind of an extra left hump or tail on the distribution, which we think means that there's some loss of function variants present in this pool. So we're currently sequencing these, flow sorting and sequencing these to figure out exactly which variants they are. I want to close with kind of a future direction, a crazy idea that we're trying to work on, which is to kind of motivated by the idea that now there's actually some, people talk about the awesome power of yeast genetics. I think there's actually some pretty awesome genetics you can do now with hex cells, with all these cell lines that exist. They're a lot easier to work with than cardiomyocytes. So what I've actually done is I've taken all the major genes necessary to set up an action potential in a heart cell, and I've ported them over and made a stable HEC293 cell line that has all of these essential components. We can do this with or without adding in that final potassium channel, which kind of stops the action potential. So here's what it looks like in a hex cell if we include the potassium channel. So we can apply a pulse of electricity to a cell, and this is using a fluorescent reporter of membrane potential. So in this case, we're seeing the negative deflections as the representation to action potential. But we get something that looks kind of like a cardiac action potential for a few hundred milliseconds, and then it repolarizes. We can apply a pulse, say every 500 milliseconds, and see a repetitive signal. If we don't include the potassium channel, by contrast, the cell depolarizes, but it basically never repolarizes and has essentially an action potential that lasts for tens of seconds, sometimes even over a minute. And turns out if you have these ion channels open for this long of a time, you get this large streaming of ions into the cell for a very long period of time. That's very toxic to the cell, and even a single pulse of electricity is actually lethal to the cells. So our idea is that we could take this cell line that we've developed and take the same libraries that we're using to measure potassium channel cell surface abundance and also put them into this cell line and get a measure of function that's pretty similar to what the potassium channel actually has to do in a cardiomyocyte. So today I've told you about a few different approaches and various stages of progress that we're taking to study cardiac ion channel function, cell surface abundance. And we're really excited that now that we have some of these methods working, we can kind of go to town over the next little while and study lots of variants over the rest of the genes. And I also want to point out that ultimately what we're really interested in is getting this data applied in the clinic, and we're thinking pretty hard about how we can use this deep mutational scanning data together with other modes that are maybe even lower throughput of studying variants, and as well as merging that together with patient data to get really accurate classifications of these variants that can be clinically used. So with that, I'd like to thank especially my mentor, Dan Roden, and the rest of our Vanderbilt team, as well as Doug and Kenny. And thanks a lot, and I can take any questions.